In this video, we will discuss about the final figure that is presented in the paper that we are discussing, where they find out the mechanisms that might cause these Cancer-Cachexia behaviors. And in general, it's the inflammation that mediates apathy-like behavior in the Cancer-Cachexia. So, in the previous video, we started to find out mechanisms, but we have not finished in the previous video. So we will continue our discussions about these mechanisms to find out the behavior in Cancer-Cachexia. So what they did is, this time previously we find out that it's the GABA receptors that cause this effect or helps in this Cancer-Cachexia behaviors. So in this case, what they did is they knocked down in these GABA receptors and the interleukin 6 that cause this effect of this Cancer-Cachexia behavior. So in this panel A, what they did is they knocked down this ARP and also this interleukin 6. And when they knock down this interleukin 6, when they knock down this interleukin 6 in the NAC, what they are able to see that the percentage leaving is upregulated compared to the tumor case. And in the panel C also, we can see that when the interleukin 6 is knocked down, we can see the percentage back point is recovered to some extent compared to the tumor case. And here is the image where we can see that in the bottom side of the panel A, where we can see that the f-cos formations are high in the case of when the interleukin 6 is not knocked down. But when the interleukin 6 is knocked down, we can see that f-cos formations also goes down. And next, here they give the informations about this GFRAL. Here we can see that f-cos formations, GFRAL and interleukin 6 receptors are shown here, and the quantifications of this is shown on the right hand side of the panel D. So far we have discussed the knockdown of this interleukin 6 from this NAC, because in NAC, when they checked about its cell surface expressions, they are able to see that NAC has more interleukin 6 cell surface expressions. Next they move to check the effect of this ARP knockdown or PBN knockdown, that is shown in this panel A. In the panel E, it is shown in detail. It is ARP and PBN knockdown, as these seems to be the initiator for triggering this these Cachexia behaviors. So when they knock down this ARP and PBN, what they are able to see is that in the case of tumors, the ARP PBNs is shown here, and the ARP PBN is knocked down, they see less, and they knock down using this DTR. So we can see that knockdown is successful, as the less amount of ARP PBNs can be seen here. And when they check the percentage breakpoint, they are able to see that with the ARP PBN knockdowns, the breakpoint is again recovered to some extent. So here they have knocked down this ARP PBN using this reagent, that is DTR. And in the panel H, what it did is they knocked down the ARP PBN using the virus, AAV virus, which has these GFP reporters. And in that case, what they are able to see that when this ARP PBNs has been knocked down, they are able to see this recovery. They are able to see the recovery of these dopamines to some extent, and from this ARP to PBN stimulations. But when this ARP PBNs is not knocked down, they can see Cancer-Cachexia behaviors. So these Cancer-Cachexia behaviors can be recovered if this ARP PBN has been knocked down, because what happens is that ARP PBN will then go to the BTA to suppress this dopamine expressions, and when they are knocked down, the suppression of dopamine expression does not occur. The complete complete pathways that they are able to find out is shown in this panel I, where we can see that when the interleukin 6 level goes down, the ARP gets area post-traumatic gets triggered, it will form this glutamate that will interact with this PBN, and it will get activated, and the GABA receptors get activated, and in return the GABA receptors will suppress the dopamine formations by the VTA, and this will cause Cancer-Cachexia behaviors that is shown in this, that is reflected in the NAC with high we can say interleukins expressions. So this is the pathways that they have finally find out based on their studies that they have presented in this paper. If you need to understand more about their experimental results in detail and wants to have a look to the supplemental figures, you can, or supplemental figures and results, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.